Hello and a warm welcome to everyone to today's webinar on Advanced Analytical Characterization Techniques for Biopharmaceutical Development, Establishing Biosimilarity. My name is Bhavika Maheshwari and I represent Veeda Group. Before we begin today's webinar, I would like to first introduce Veeda Group to you. Veeda Group is a global integrated and technology driven contract research organization committed to delivering excellence in everything from preclinical to clinical trials and bioanalytical services. We provide comprehensive end to end solutions to innovators, biosimilars, and generic drug development programs for our global clientele. With sites across 26 geographies, Vida Group serves as a trusted partner to our clients worldwide. Together, we cater to pharmaceutical and biopharmaceutical industry, agrochemical and industrial chemical industries, herbals or nutraceuticals, and medical devices industry. At Vida Group, our focus on excellence in everything drives us to continuously innovate in delivering cutting edge solutions and uphold the highest standards of quality and reliability. Our advanced infrastructure, strategic global partnerships, and client centric approach has positioned us in a leading and accelerating journey from drug discovery to impactful healthcare outcomes. As your facilitator today, I would like to introduce you all to a couple of pointers before we begin the session. This session will have a presentation in the beginning and that will be followed by a Q&A round. We have enabled ask a question features. You can drop all your questions related to the webinar there. Moving forward, I would like you all to introduce our panelist for the today. First, we have Mr. Nandisha NP. He is a characterization specialist at Vida Biopharma. He brings over 5 years of experience in the biopharmaceutical industry. He's got a master's degree in biotechnology. He leads the primary characterization, where he's employing advanced techniques such as high resolution mass spectrometry, HPLC, UPLC, and CE. His expertise spans in oligonucleotides, peptides, insulin analogs, fusion proteins, and monoclonal antibodies. Mr. Nandisha is playing a key role in ensuring the quality and reliability of analytical data, and he's fully supporting Vida Biopharma's commitment to advancing innovation and excellence in biosimilar development. Now, let me introduce to our second panelist for today, Dr. Shoman Saha. He is an associate research scientist in analytics and characterization department. He at Vida Biopharma, he is specializing in high order structure characterization. He's got a PhD in biophysics and molecular spectroscopy. He brings over 6 years of expertise both from academia and industry. At Vida Biopharma, Dr. Shoman Shaha leads the structural analysis of proteins, peptides, and monoclonal antibodies, where he's using techniques such as circular dichroism CD, FTIR, DLS, and DSC. He is also utilizing advanced technologies including high resolution mass spectrometry and SEC, FFF, MALS to further enhance and refine our analytical characterization process. So, without any further delay, let me virtually call upon Mr. Nandisha to begin the session. And then over to you, sir.
Hi Bhavana, thanks for the introduction. Hi everyone, nice to meet you here. So thanks for joining us today. So today, we'll have the presentation as Bhavana mentioned. So followed by QA questions will be there. If anyone have any question and answer, you guys can type it in the chat box. So without further delay, so we'll start the presentation. Today we are here to discuss the advanced analytical characterization techniques in the biopharmaceuticals development, establishing the biosimilarities. As we know, analytical methods are so crucial for that all these biosimilarity assessment on the biopharmaceutical development. So in this webinar, particularly we are going to discuss what are the advanced techniques. So we are using to make the in the form of like biosimilarity assessment to make the good data on all these things. Next step, please. Yeah, first let us start with the basics. So what is first, like we'll talk with what is biologics and biosimilars. So first, like biologic is like are the complex large molecules. So made of, as the name suggests, it's made up from like living organism. These product biologics are the products, first kind of drug products which will be developed in specific conditions, and these are like protected by patenting. And next, like example, like trastuzumab, pertuzumab, and like emicizumab, all these things. And next is biosimilars. Biosimilars or the biosimilars are like, as the name mentioned, is like it's also like originated from the living organism, and these are like similar version of the biologics. So like in terms of like structure, analytical structures, and functional structures. So like what are the key features of like similarity between like biologics and biosimilar, that we'll discuss on the next slides. Example, like why we are, yeah, mainly why we are looking, people are looking into the biosimilarities when we have that already biologics. So when the people are developing biosimilars, mainly because of the two things. Like one is like a reference product is like like it's already protected by patent, and then availability of the like drug, and the cost reduction. So by keeping all these active thinking, like people are looking into biosimilarities. And if you see that geographically, like now how the biosimilarities is evolving today. So like till now, like from 2015 to 2024, almost 62 biosimilars are identified, and like it is like patented now. Okay, from if you see from last this like 2024, so this currently this year only it is like already 17 biosimilars are like what is like identified, and like they are in the market. So now let's go like what is it exactly that that biosimilarity workflow, how it will take, what are the things. So as we mentioned, like biologics is like here, if, just consider as like biologics is the reference compound. And then we'll start discuss like whatever in that right side, if you give like what is the totality of the evidence for that biosimilarities characterizations. So if you see in the reference product characterizations, so analytical is the base for all these characterization thing, like whether that molecule is like what is the molecular weight, what is the nature of the molecule, how is like analytical structure of the molecule, analytical activity, and functional characterization to check the biological activity. So and then it is followed by non-clinical studies and clinical studies, clinical pharmacologies and clinical studies, and then it is approved, and then they will submit for that license. They will file, they will check for that what is it, file the license in the regulatories, and they will get the approval. That is like these are the workflows. So for the reference product, when it is come, if you see in that reference product characterization, the analytical part is like most basics, and it might play like only that smaller size of like portion. But when it is come to biosimilarities characterizations, so analytical characterizations play a major role, as we are indicating in the image in the right side, stepwise approach to increase the certainty of the molecule. So analytical characterization of the biosimilarities play a critical role. Next slide. Yeah. So when we are looking into things, what is the role of analytical science? So when it is come to analytical science, here we can divide it into two things, like product like, it's like in the product characterization we can divide into two things, that is like primary structure and higher order structure. If it is come to primary structures, we can tell like what is the molecular weight of the things, what is the amino acid sequencing, what is like disulfide linkage, what is the post translational modifications is present in the molecule. So all these kinds we can studies in the like primary characterization. In HYS, if you come, what is the secondary structures, what is the tertiary structure of the molecule, even including quaternary, and then what is the stability of the molecule. So all these things we can check in the things. And then this will be like regarding the product characterizations. And when it is come to analytical CMC may we can identify like what we can do that identification of the molecule, and with like with our analytical techniques, and we can check the purity of the molecule, how much purity is there, and then if there is any heterogeneity in the molecule, like any multiforms is available in the like purified molecule, that one, or any variants is there in the this one, we can check. And whether like in the variants means whether there's charge variant or size variant, anything, or in the residuals, if it is come to like whether any process related any residuals are there or not, that we can check in analytical CMC. And followed by potency will be taken care by functional characterization. Next slide please. Yes, when we are talking about like analytical characterization of the molecules, we need to consider like what is like critical quality attributes we called as like CQAs. So what are the CQAs will comes into picture? CQA like is divided into four categories like primary structures, process and product variants, uh, process modification, identification, quantification, and HYS part. As like I am the characterization specialist, like we will talk about like we will explain. We'll start with the primary structure characterization. So if it is come to primary structure characterization, what is the intact mass of the molecule? Of the molecule means like whole molecular weight of the molecule, or and subunit mass analysis means like if there is any subunits are available. Example, if you are talking about like all these like monoclonal antibodies, like heavy chain, light chain, so how it is like how the like how those things are arranged, and all these things we can study there. And then if the next one is like peptide mapping and sequencing. In the peptide mapping and sequencing, we can study like whether the coverage, like whatever we are have like amino acid sequencing, whether all the amino acid we can check like amino acid sequencing is like it's like arranged is properly or not. And then like any post transfer modification is available, and any terminal sequencing, like we will do the terminal sequencing, like any usually what will happen like when we are talking about is all this like monoclonal antibodies and any biologics and biosimilars. So at the end they have like N terminal and C terminal sequence will talk. So those are the endpoints where they get into more prone to get modification. So those modification also we can study in the peptide mapping and the sequencing. And next is like process and product variants. If it is come to process and product variants, so we can check like whether size and charge variant means like whether when they will do all this like production of all this like monoclonal antibodies upstream and downstream process, and they will do purification when they will submit for the analytical characterization, whether we can check like whether that molecule like how the size variance is available, whether like it is monomer or dimer, tetramer, all these things we can check. And then charge variant, like whether the molecule have like cation or anion based on the charge, so that also we can study. And then it's like host cell residues, like when they will do like upstream and downstream process, that is what is the host cell, they will choose the host cells. So whether any residuals is available in it came from that host cell or not, that also we can check. And then impurity characterizations, so we know impurities are like will play a big role in the like all the products, because these are the things like which will make the sample like impact on the sample safety, efficacy, activity of all the molecules. So that also we can study in like process and product variants. Then if it is come to PTMs, identification and quantification, so PTMs are like most crucial for that product, which will give you like oxidation, demidation, and then glycation. So release glycan analysis. So these are like will help the product to no in like product efficacy, structure, stability. All these things will support. And then it is like HOS. HOS may like as I mentioned in the previous one. So we can study the secondary structure of the molecule, tertiary structure of the molecule, and conformation stability of the molecule. All these things we can study in the HOS. Next slide, please. Yeah, when we are going to study them in biosimilar assessment, what are the main criteria we need to like think? Like what are the main criteria are available as per guidelines and all these things. So if you see like in our this on left side we were like in biologics we kept, as we know that biologic is like innovator drug, and a biosimilarities is like the no like which is going to mimic that no, innovator means biologics, and a biobetter is like similar to that biologics which will be more better than that biologics. So this we call as biobetter. So like when it is come to the biosimilar assessment, so like how to do the biosimilar assessment, like what how many batches we need to do and all these things. So if you guys is like if you are taking one batch of biosimilarities versus one batch of like biologics, so this from this we can say only like critical structure attributes, that is like peptide mapping, molecular weight analysis, and a CAF and N-glycan analysis, and SEC mals and CD. So if you do CD, we'll get like second and tertiary structure. If you do SEC mals, we'll get like what is the molecular weight of the things based on the size. And if you do an early and glycans, like what are the types of glycans, glycoforms are available in the molecule, that we can study. And CIF, what is like PI of the molecule, isoelectric bond, that we can study. And then we'll do only like I know like molecular weight analysis, what is the molecular weight of the things. And peptide mapping we can study like what is like amino acid sequence and post transition modifications. If you do one versus one. And then if you are considering like three batches biosimilar against the three batches of innovators, so that will be good for like all the analysis of this thing. Biosimilar assessment is like whenever we are doing any biosimilarity assessment and analytical techniques, so there is a guidelines called like ICH guidelines, Q6B characterization. These guidelines specifically tell us to use the minimum 3 versus 3 to get the full ICH Q6B characterization. And we can do 3 versus 3 and 6 versus 6 and more comparable studies. And when we'll do more like comparable studies, more batches and more studies, that will give more accurate data. For them this will be good for that complete characterization of the product. Next slide please. So I'm being like in Veeda, so to do all this, like I am the characterization specialist, especially primary characterization specialist. So here, if it is come to primary characterization, we have LC, HPLC, LCMS, and micro CE and SDS page. So these are the four components which will be most critical for the primary structure characterization. If we start with like LC, so in LC, I mean like HPLC, liquid. High pressure liquid chromatography. So this will be like from this we can we can check the amino acid composition analysis and purity by RPHPLCs. And like this will purity say we will do to check that whether purification in the upstream and downstream processes happen properly or not. And like we'll do that IEX by HPLC to check like what are the charge variant forms are available in the molecule. And then followed by like SEC HPLC, which will give like how much like what are the different size aggregates are present in the molecule. And then proteome analysis by Hillic and HISM is hydrophilic and hydrophobic interactions with that. And then if it is come to LCMS, as we know, LCMS is like liquid chromatography combined with like mass spectroscopy. So this will give most of the primary things, primary structure supporting data, which will be like intact or native molecular mass, means like what is the exact molecular weight of the proteins or any hormones or any like fusion proteins and all. So this will be support for it in intact and molecular mass. And then subunit mass analysis, what are the different subunits are available in the proteins, that one we can study. The peptide mapping, whether like whatever we have like amino acid composition that is in the proper this thing like an arrangement or not, that one we can study. And then peptide sequencing analysis for coverage. So where we will do the peptide mapping, that will be what will like this will be like protein will submit the things into the reduction, alkylation, followed by enzymatic digestions. So which kind of enzyme we need to select here to get the sequence coverage. So from to get the peptide sequence coverage like 100% or 95%, whether like sometime we will use a single enzyme, sometime we'll use a dual enzyme to get the like most to cover the most of the amino acids. And then disulfide mapping analysis, as you know, disulfides plays a very big role in like for the structure and stability perspective of the molecules. To we will study that also. So here in the if you're planning like disulfide mapping analysis will be directly like denature of the molecule, and followed by like enzymatic digestion. From this we'll come to know the what are the like disulfates are like from where to where it is linked, so whether it is intact or not, then all those things we can study in the things. So and the glycan analysis like we can glycan analysis, we know these are the like sugar moieties. These are will be very good, like play a key role in the for the structures and stability of the proteins. So like if the glycans are attached to aspargine, we'll say that is n-glycan and n-glycans, and if the glycans are attached to threonine and all, we'll say like o-glycans. These analysis also we'll do in our facility, and we have developed a kit-based methods for this thing. And then variant and impurities, like we can do that impurities analysis, and if there's any variants are available or any impurities in the molecule, that also we can do. And then we have micro CEMS, which will be miniature version of that MS, where like in the volume and the time will be reduced compared to MS. And then SDS page as we know, like yeah, gel electrophoresis traditional method for us, like to check the PI of the molecule. And like if you're talking about like we can do that, native SDS page, native page and SDS page, 2D gel electrophoresis and Western blotting to check the antibody specificity. And then we can will perform the PI and CESDS and CZD, CIF. These are the primary structures tools available in the Veeda. We have expertise in that for these things. Next slide, please. Yeah, let's take a monoclonal antibody as an example. So now here we'll go through that individual as like how we were doing step by steps, and like what are the primary like as we are talking about like characterization of like is the if you take like MAB as a example. So characterization of MAB, what are the things methods we are doing and how we are doing. So that we will talk. For example, let's talk intact map, like we intact map and subunit will do to check the whether like molecular weight of the protein maps and subunit of the maps. And then followed by peptide mapping sequencing, glycopeptides are like peptides which is attached to the glycans that we will analyze. And then if this come to post translational modifications, we can study like post translational modifications or the modifications which is occur like during the process or the shipment, all these things. So then what are the those modifications are occurred, these modifications will play a big role, because like if there is some modification changes at the if there's modification occur in the proteins or amino acids, so then that activity of the amino acids or the proteins that will be changed. So then what will happen, all those things we can try, we can check here, and we will do the things like example pyroglutamation, oxidation, deamidation, terminal lysine clipping, or if any truncation is truncations are there in the molecule or not, and many more. And then terminal sequencing, as I in the previous slide I mentioned. So these are the terminal of the monoclonal antibodies or any proteins which are more prone to get modified easily, or to we can study that whether any modifications is occurred in the terminal sequencing or not, from the like N terminal and C terminal sequencing. And then disulfide mapping is like disulfides are the bond, covalent bond which will be connect to cysteines or cysteines. So whatever we can study the nature of the disulfide bond, whether they are in the native form or the scrambled form. If the scrambled is happened, what are the impact that will happen, like we know if the disulfide bond scrambling is happened, that will make the drug activity, it will reduce the drug activity, and it will be impact on the drug safety also. So all these things, that thing we can study in the disulfide mapping. And release glycan analysis. So in the release n-glycan analysis, so we can study what kind of like what are the glycans are available in the molecule. So whether that G0, G0F, G2F, and whether manospaces are available or not. So if the manospace is available, how much is the percent of the things. So what will be impact on the manospaces available, manospaces which is available in the molecule. All these things we can study in the n-glycans. And next is like size variants, next chapter. So like in the size variants we can study whether the that size are oligomers or aggregates, or in the next is like charge variants. The means like whether the whatever the charge variants available in the molecule, this one proteins, whether like those are acidic charge or basic charge, and what kind of amino acids are available in that. And then we can study the secondary structure, tertiary structure analysis, and then we can study the intrinsic and extrinsic fluorescence also to know the molecule nature. So these are the typical what is it, analytical characterization tools which we'll use for that maps. Okay, so this is one example. So next we'll start with like individual chapter. Next slide, please. Yeah, let's take an example of molecular mass. So when we'll do it like in the lab, like what is that, intact and subunit. Intact is like to study the whole molecular mass of the protein, and subunit mass like whether like to study whether any modifications occurred in the like light chain and heavy chain. That one also, because like for example, sometimes we have like fusion or like bispecific maps. So that time we'll get a difference between like normal maps and a bispecific map. That also we can study in that. So from like for example, if you see in the right side of the slides, what are the things we can get from the intact things, like molecular mass of the intact protein will get in that. It include like unmodified proteins, means like protein is like without modification as such. Once we purify the molecule proteins, what is the state, that one will get. And a glycoforms, like so when we'll do the intact along with the molecular weight, we'll get the glycoforms which is attached to that molecule. So that one we can study in this. That is like example G0F, G1F, G2F, etc. These are the things. And is there any truncations is available? Truncation forms are available in the things. Mainly this will happen C terminal of lysine. So particularly that we can study in the things. And then it's come to molecular mass of the subunit protein. As I mentioned in the previous, from this we can study whether like light chain, heavy chain, or uh, glycoforms of all these things, and truncation forms. And sometimes what will happen when the processing like all the upstream process and downstream process are happened, and due to some changes in the conditions, we will get like what will happen. We know like maps are like two heavy chain and two heavy chain and two light chains will be available. So if there is any breakages in the light chain or heavy chain or any modification in that, that also we can study by FAB and FD things. And if you come to like left side of the things, what are the things we will do in that experiment wise. So in the intact, just directly we will inject the protein as such protein into the LCMS. So means like what you call it, we will get the molecular weight of the protein. And if it is come to reduce mass, so what will happen in to reduce mass, as I mentioned, this is to study the light chain and heavy chain. So here, so as we know, light chain and heavy chains are linked with a covalent bond which is called as disulfide bonds. So these disulfide bonds will reduce with the reducing agent, and then we'll submit the samples to LCMS, which will give the reduced mass data of like light chain and heavy chain. That one we can see in the presentation. So if you guys see in the light chain we are have on this one, and heavy chain, what is the chromatography things we will get, that one we can show here. And then if you want, if there is anything, we can do the IDES digest to study that subunit and all. So these are the experimental things what we can do, what we are doing in the this thing. And in the Veeda Biopharma, we have expertise in monoclonal antibodies, fusion proteins, and glycosylated proteins, peptides, biosimilarity comparability and assessment. So actually we have handled like almost like more than 10 plus maps, and two plus fusion proteins, and highly glycosylated proteins, and normal peptides and insulin analogs. For all these things, we are doing biosimilarity, comparability, and assessment. And next, we will move to the next chapter. Next slide, please. Next chapter is like amino acid sequencing. So in the amino acid sequencing, we will do peptide mapping and its sequencing. So what we'll do in the peptide mapping, we will take the proteins, we will do the denaturation of the protein, because the proteins, if the proteins is highly glycosylated, then followed by will reduce by reducing agent, and alkylate with alkylating agent. And all after this one, we'll submit the reaction into the enzymatic digestion, which will be the particular enzyme will be choose based on the amino acid sequence. That enzyme will go and cleave at particular peptides. So then we'll get like 4 to 5 amino acids each peptides. From that we can study the peptide mapping and sequencing. So that is the thing. And in the left side, if you guys see, this is the or the like data chromatogram what we'll get in the MS. This is we call it TIC, means total ion chromatogram. And then if by doing this we will get peptide mapping and peptide sequencing. From this peptide mapping, what are the things we'll get? So from that we'll get like molecular mass of the peptides. For example, if you have any signature peptide to identify the protein, that one will get. And retention time and mass accuracy of the peptides, and glycopeptides identified, as I mentioned in the previous one while talking. So glycans which is attached to the peptides, so like that one will call as a glycopeptide, that also we can identify it. And PTM's peptide identification, so if there in the peptide any post-translational modification is available, that also we can check. And we can quantify the peptide, quantification of the peptide also we can do. So this is thing for the peptide mapping. And then if it is come to peptide, the peptide sequencing, so as we know, when we use it enzyme, it will go and cleave at particular site. For example, trypsin, lysine, and arginine. And if you use gluci, it will go and cleave at glutamic acid and aspartic acid. So when the digestion is happened, that time we'll get the fragment of peptides. From that, we can check we can do the peptide sequencing. Like that peptide itself, we will do the sequencing with that Bi and Y ions. That will give the confirmation of like that is a peptide exactly what we are looking, and this is the mass accuracy of the thing. So after that, we'll get like amino acid sequence coverage. And from this we can tell like fragmentation optimization also we can study. And we have expertise by achieving 100% sequences by using either single enzyme or dual enzyme or multi enzyme. Like sometime if the molecule has like different amino acid sequence, so then we will use two enzymes like gluci, example gluci or and trypsin. Or sometime only one enzyme may be sufficient. For example, if you are using any insulin molecules, so gluci alone will give like for us like 90 more than 95% of the coverage. And if you are using maps, so trypsin one, a single enzyme is sufficient to get the like more than 95%. But however, when we do the sequence coverage uh, in the maps, we have GEC peptide. So that to cover, we need the what is it gluci enzymes. So that will contribute the terminal sequencing. This is the way when we will select the enzymes. So by choosing by seeing like what is the amino acid composition available in the molecule. From this we'll complete we will complete this amino acid sequence chapter. Next we'll move to the next chapter. So next one is most important thing is like cysteine linkage. So usually we'll talk about disulfide bond and disulfide linkages. Okay, these disulfide linkages are like cysteine linkages, that these disulfides are formed between cysteine and cysteine. That's the reason here we are calling as a cysteine linkages. So why we will do here like native disulfide linkages or like by doing the disulfide linkages we will study the native disulfide linkages, or like scrambled disulfide linkages, or like how it is arranged. So for example, if you see like as we know maps, there are four types: IgG1, IgG2, IgG3, and IgG4. So these are like no different there are composition is different, and few are highly glycosylated and few are less. And the disulfide, if you see, IgG2 has more disulfide bonds than like other. So IgG1 and IgG3 and IgG4, like these are the difference as in the like key and the key differences in the this thing like disulfide things. So when we'll do the disulfide mapping, so from that we can study the native disulfide analysis. From that we can study the conformation of native no, cysteine to cysteine linkages from non-reduced profiles. So means like when we are doing this disulfide linkage in the experimentally, what we will do, we will denature the samples, and followed by we'll directly will submit the sample into the enzymatic digestion without reducing the disulfide things. So that is the name here mentioned. So from that we can know like in a non-reduced profile we can study the cysteine linkages. And orthogonal confirmation using the reduce and alkylated profiles means like when we submit the things, when we submit the molecule, so to that reduce alkylated followed by enzymatic digestion, from that that is orthogonal conformations. So using like to confirm that to reduce and alkylate profiles. And then we can study the scrambled disulfide analysis. So that is like to search like any modification is and like mismodification. Like for example, we are thinking like from this amino acids to this amino acids there should be like disulfide bond linkages, but by mistakenly during the process or anything, if it is like that one is instead of this cysteine, if it is no go and like form the disulfide with other cysteine, what will happen? That is scrambling of the cysteine bonds that we can study in the like in a scrambled disulfide analysis, and then we can identify and confirm the scrambled disulfide linkage peptides. So usually when we'll do like native disulfide or the scrambled disulfide analysis, so these disulfides are intact. Okay, this will give some peptides that from that peptide we can check that, so we will get the confirmation of scrambled disulfide. So, and being in Veeda as like being like a primary characterization specialist, we have expertise in disulfide linkage analysis in the maps, proteins, cyclic peptides, like example linear and like we have biosimilarity, comparability, and assessment experience. From this, we'll complete a cysteine linkage chapter. Now we have another one more chapter which is most crucial and last this thing. Yeah, this is the current chapter is like release glycans. So glycanalysis, which will be most important and crucial for that to study about this to study of these things. So because so these are plays a key role in the structure, safety, and efficacy of the molecules. So if you are talking first, we'll talk about like experimentally what are the experimental things we will do for the glycanalysis. So when we'll take the maps, so we will do the denature followed by the release N glycans with PNGSF enzyme, and label the glycans. So with the rapid flow MS labeling reagent or 2AB, these are the labeling reagents available in the market we can use. And then once the labeling is done, so these will be purified with the Hillick microelution plates, which like means Hillick principle. Okay, this is like experimental part. So what we'll do when we'll do this, so we'll annotate the IgG N glycans with like with fluorescence based N glycans identification. So that may be 2AB or rapid flow MS, all these things. So these are like MS based identification and like means like these are like mass based. So when we do in our Vida facility, we are doing like all these like mass based and glycan identifications. So what will be the future? What are the things when we characterize of IgG N glycans? So what we'll do, we'll identify the unknown N glycans and we'll do MS and MS for unknown N glycans to characterize things to confirm like what is like mass based identification of the things. So and then when we identify identification and characterization of O and N glycans, so when we these are like this will be mainly like as I mentioned in the experimental part, these are like chemical and enzymatic based O glycans release identification and characterizations. And in Veeda we have expertise in like establish the method for the release of N glycan analysis for the monoclonal antibodies and the glycoproteins we supported with NIST map standards for the system method and for system and method suitability. Also, we have the experience in handling like O glycans. So hi, here like till now I explained about primary characterization structures. So what are the tools available with us and what are the experimental things we are doing in our Vida facility. Now I will like to call my colleague Dr. Sauman Shah. He is expert in like HOS, so he will take care of the coming up slides. Thank you.
Hello everyone. Thanks Bhavika for your kind introduction. Thanks Nandish for your insightful and informative speech on biosimilars and primary structural characterization. As we know, the success of biosimilars relies not only on demonstrating efficacy and safety, but also on ensuring that their molecular structure is nearly identical to that of the reference biologic. Specifically, understanding the maintaining of the higher order structure or HOS of protein is very crucial for ensuring that the biosimilar has the same therapeutic properties and minimizes immunogenic risks. So in this slide, I'll tell about the introduction about HOS. So in pharmaceutical industry, biopharmaceutical deals with a wide variety of large molecules, including monoclonal antibodies, antibody drug conjugate, fusion proteins, biospecific proteins, etc., which are known for their huge size and complexity. These molecules consist hundreds or even thousands of amino acid residues, which intricate folding and high complex 3D structure. Let's start from the preparation of a protein. The manufacturing process plays a critical role in the determination of the final structure and function of a protein. This includes the choice of host cell system, the expression conditions, and purification methods. Each step in the process can introduce variation in the protein structure that may affect in biological activity. Additionally, purification methods can affect the protein's final conformation. For example, using harsh detergent or incorrect pH during purification can also lead to protein denaturation or aggregation. After process, we get the final product. But the final product itself, the protein in its form, drug-ready form, must retain a precise structure for effective and safe therapeutic use. Even small changes in protein structure, such as misfolding or aggregation, can lead a significant alteration in function. For example, proteins that are not properly folded may fail to bind to their target receptor or enzymes, rendering the therapeutic inactiveness. So now coming to the formulation stage, which is the final step that ensures the protein remains stable and effective until it is administered to patients. The formulation of biologics includes the choice of excipients, stabilizers, and buffers, that preserves the protein structure and stability. For example, proteins are often formulated with stabilizers to prevent aggregation or denaturation during the storage. So many things we are seeing. So in process stage, in the product ready stage, and formulation stage. Now the question is how to ensure correct structure of a protein or a biologic or a biosimilar. So moving to the next slide. So in this slide, I'll be talking why HOS is critical for biologics and biosimilars. See, my colleague Nandish already told about the biologics and biosimilar. Now I will speak why HOS is very critical for the biologics and biosimilar. In biologics, we talk about new biological entity. It can be monoclonal antibody, it can be antibody drug conjugate or fusion protein. So many things are there. These structures are so large and complex in nature. Their 3D structure is intrinsically linked with their function. So example, in monoclonal antibodies, the binding of an antibody to a specific antigen is highly dependent on the precise conformation of the protein. For biologics, both discovery and development are required. So any step, if the higher order structure is altered, even by a small amount, it can result in loss of activity, reduced efficacy, or even unwanted immune reactions. In biologic drug development, ensuring that the protein's higher order structure is preserved throughout the production, formulation, and storage is essential. Variation in temperature, pH, or other environmental conditions also can destabilize the protein, leading to denaturation, aggregation, or the formation of non-functional isoforms. This is why HOS is considered as critical quality attribute we call CQA for biologics, and its directly effect on the function of the drug. Why HOS is critical for biosimilars? Biosimilars are biologic drugs that are highly similar to an already approved reference biologic but are not identical, which while biosimilars are designed to be highly similar in terms of structure, function, and clinical performance to the reference product. As a result, ensuring that a biosimilar matches the higher order structure of the reference product is crucial for demonstrating similarity in both function and safety. Since biosimilars are not exact replicas of their reference biologics, they undergo rigorous characterization to ensure that their higher order structure is comparable to the reference product. Thus, HOS is key parameter in demonstrating that a biosimilar matches the reference product's structure and function, ensuring that the biosimilar provides the same clinical benefit at the benefit as the biologics to the patients. For biosimilar development, ensuring that the higher order structures of the biosimilar is consistent with the reference product is essential for several reasons. Those reasons are to match quality of the biosimilar, to ensure the stability of the biosimilar, its therapeutic efficacy, and mostly the safety, because alteration in any structure can increase the risk of immune response, making it crucial to avoid introducing immunogenic variants. Can you move for the next slide? Now, in this slide, I will give brief rationale about HOS, how HOS becomes so crucial in early stage of biosimilar development. As I told earlier slides, in biosimilar development, we deal with large biomolecules. So, some factors are coming to when we are dealing with the HOS. First, it's coming first, protein size and complexity. Protein used in biologic therapies are large and complex molecules, often consisting of hundreds or even thousands of amino acids. The structure and overall complexity of a protein can directly affect its biological activity. For example, even slight changes in the 3D structure of a protein can alter how it binds to its target molecule. And next part is the expression. Proteins in biologic drugs are often expressed in mammalian cells, such as Chinese hamster ovary cell. Commonly, we call it CHO cells. And this system can perform complex post-translational modifications. So, post-translation modifications we call PTMs, such as glycosylations, phosphorylation, and acetylation, play a critical role in shaping a protein's HOS and ensuring it remains functional. But of this PTM, glycosylation is very crucial. As told earlier by Nandish about glycosylation patterns are closely matched with the reference product. Even minor differences in PTMs can affect the pharmacokinetics, efficacy, or immunogenicity of the protein. Additionally, biosimilars must be checked for any protein variants that may arise due to slight sequence difference, and this can also impact the higher order structures. Next, moving to the disulfide linkages. Disulfide bonds are covalent linkages that stabilize a protein's tertiary and quaternary structures. Particularly for proteins that rely on multi-subunit arrangement, these bonds are essential for maintaining the correct 3D shape of the protein and ensuring the functionality. So variations in protein size, expressional system, post-translational modification, and disulfide linkage can significantly affect in the protein's 3D structure and molecular consistently and its therapeutic performance. By using advanced analytical techniques to thoroughly characterize these structural elements, manufacturers can assure that biosimilar closely matches to the reference product, providing patients with treatment that is safe, effective, and therapeutically equivalent. So in this slide, I will tell about the importance of HOS in biosimilars. Higher order structure plays important role in biosimilar development, particularly in relation to the structure-function relationship and its impact on the batch-to-batch consistency, stability, and variance. Even slight change in HOS can lead to alter in the protein's activity, efficiency, and safety. One of the most important aspects of HOS in biosimilars is ensuring batch-to-batch consistency, because in any stage of process and formulation can alter the structure and conformation of the entire batch. In biologic manufacturing, slight variation in process condition can lead to differences in protein folding, PTMs, and other overall structure. It is crucial that biosimilars maintain consistent HOS across different production batches to ensure reliable therapeutic outcomes for the patients. Additionally, stability is also another key factor. A stable higher order structure ensures that the biosimilar maintains a functional integrity during storage and throughout the shelf life. Any degradation in the structure can lead loss of activity or unwanted immunogenic response. Finally, biosimilars may exhibit variants due to minor differences in expression system, post-translational modifications, or processing conditions. These variants must be carefully monitored to ensure that don't impact on the protein functional properties or deviate from the reference biologic. In summary, the HOS of biosimilars must be carefully controlled and characterized to ensure that the protein performs as expected. This is critical for ensuring consistency, stability, and therapeutic equivalence across batches and ensuring the safety and efficacy of the product for patients. Moving to the next slide. So in the last few slides, I spoke so many things about HOS. Now I will tell what actually HOS is and what are the components of HOS. So when we talk about higher order structure, we refer to the three-dimensional arrangement of atoms in a protein, which directly influence its functionality and efficacy. This higher order structure, which can be secondary, tertiary, or quaternary, each play a role in protein's overall biological activity, stability, and immunogenicity. Let's take a moment to review these structural labels. So let's start with secondary structure. The secondary structure refers to the local folding of the polypeptide chains into structure like alpha helix, beta sheet, beta turns, and random coils. These patterns are stabilized by hydrogen bonds and are critical for the protein's overall fold and stability. Any significant alteration to the secondary structure can compromise the protein stability, activity, and potential for aggregation. Now, moving to tertiary structure. Tertiary structure is the three-dimensional folding of the entire polypeptide chain, which result from the interaction between the amino acid side chains. This includes functional groups, disulfide bridges, hydrogen bonds, and different interactions also. This structure determines the functional properties of the protein, including its ability to bind to the target molecules like receptors or enzymes. Next is quaternary structure. Quaternary structure refers to the arrangement and interaction of multiple protein subunits in a multi-subunit complex. In biosimilar, if the reference product functions as multimeric protein, maintaining the correct quaternary structure is crucial for ensuring that the biosimilar exhibits same functional properties. Along in the secondary, tertiary, and quaternary structures, conformational changes also play a crucial role in HOS characterization. Conformational changes in proteins refers to the alteration in their three-dimensional structure affecting their secondary, tertiary, and quaternary structure. This can impact on protein function, stability, and interaction with their molecules. So now, to ensure that these higher order structures are maintained, several analytical techniques based on spectroscopy, calorimetry, and light scattering are employed in our facility to carefully characterize the HOS of the biosimilars. Let's explore some of these critical methods. So first of all, I'd like to discuss about CD, circular dichroism. So CD is one of the most widely used techniques to study the secondary and tertiary structure of proteins. Far UV CD measures the overall folding of the protein and give us the content of secondary structural component like alpha helix, beta sheet, beta turns, and random coils. Whereas near UVCD can measure the tertiary structure, including interacting amino acid residues like tryptophan, tyrosine, phenylalanine, along with the disulfide bonds. But by comparing the CD spectra of the biosimilar and the reference product, we can verify whether their secondary and tertiary structures are similar, which is very essential for ensuring that the biosimilar will have comparable stability and activity. Now, moving to the Fourier transform infrared spectroscopy, we call it FTIR. FTIR spectroscopy is another crucial tool for investigating the secondary structure of protein, especially for studying amide 1 and 2 bands that are associated with proteins folding. FTIR can give insight into the protein's backbone conformation and allow us to identify the differences between biosimilar and the reference biologic, ensuring the higher order structure is preserved throughout. Next, coming to the size exclusion chromatography. field flow fractionation with multi-angle light scattering. We call it ACC-FFF combined with MALS. So size exclusion chromatography combined with MALS, multi-angle light scattering, is an excellent method for evaluating the size, aggregation, and overall structural integrity of proteins, providing direct information about the size distribution and oligomerization state of the biosimilar. Field for fractionation, or FFF, is an extension of this technique, which can separate the particle based on shape, size, and density. And for even larger particle and larger aggregates, we can analyze with these techniques. Now moving towards the differential scanning calorimetry, DSC. So in DSC measured the heat change associated with the protein folding and unfolding. It is particularly useful for assessing the thermal stability of the biosimilar and the reference product. And the next one is DLS, dynamic light scattering. Dynamic light scattering is another widely used technique that measures the size distribution of particles in solution based on the light scattering of particles in motion. This technique is especially useful for assisting the size and aggregation of protein, providing real-time information on any variation in particle size aggregation and any tendencies between the biosimilar and reference product. So moving towards the next slides. So now coming to this typical development path in biosimilars for HOS monitoring. Here I'll be telling how one HOS characterization happens in this biopharmaceutical industry. Let's consider with maps, because maps we are using every day for the pharmaceutical in biosimilar and biologics. So considering map, I have and already Nandish also told so many things about maps and maps primary characterization. So for preparing an innovator or a new biological entity, the manufacturing team use some process, analytical development, and formulation development. But while preparing the biosimilar, this team is completely unaware about the techniques that had used for the preparation of innovator. So it is entirely a challenge to them, challenge to the team who are preparing the biosimilar to match the structure conformation of the biosimilar with the biologics or with the innovator molecule. So in next step, so in the first step, they will start with the process, then they will move to the analytical development, or then next they will move to the formulation development. So in any of these steps, whether it can be process or analytical development or a formulation development, if some alteration occurred, the biosimilar will not match the structure of innovator, which will affect the function and the activity of the biosimilar. So to ensure the product quality assessment of the biosimilar, HOS characterization should be executed. Now, after HOS characterization, the team should evaluate the results. If alteration found related to the products related problems, such as post-translational modifications, some variants, so then the process team will be informed. And if sometime, if any case, in HOS evaluation, it is found that it is formulation related issue like stability, aggregation observed, then the formulation team will be informed to rectify the concerned issues. If all the HOS parameters found nearly identical to the innovator, then the biosimilar will be sent to the preclinical studies for the toxicology assessments. Now, next slide, please. So in this slide, I'll be discussing about the governing factors of HOS. There are so many factors that can impact on HOS, starting from the oxidation, which can impact on the conformational stability, functional stability, thermal stability, and aggregation also. Coming to deamidation. Deamidation, which is a kind of post-translational modification, can alter the conformational and functional stability. Another PTM, glycosylation, it will affect on conformational and functional stability as well as aggregation and solubility of the product. pH is also very much important factor, as it can alter the conformational, functional, and thermal stability as well as aggregation. Temperature can also differs the conformation and thermal stability as well as aggregation, leads to aggregation. Ionic strength also affects on the thermal stability. So in HOS characterization, these key factors should be taken care to ensure the quality, stability and efficacy of the biosimilar. Now moving to the regulator perspective on HOS. Next slide, please. In ICH, which is International Council for Harmonization, their guideline ICH Q6B, which is specification, test procedure and acceptance criteria for biotechnological and biological products, they clearly tells to ensure the HOS of the biological product. Another HOS ICH guideline Q5E, which is compatibility of biotechnological or biological products subject to change their manufacturing process. It's mentioned that the manufacturer should attempt to determine the complete HOS including secondary, tertiary and quaternary structure. So from the ICH guideline, it's clear that how important is HOS while characterizing or while preparing one biosimilar. So next slide. So in this slide, it will tell about the quality consideration in demonstrating biosimilarity of therapeutic protein with reference product. In this guidance also, it is clearly mentioned to executing HOS parameter in order to release the biosimilar in the market. So from these ICH guidelines and all these guidance guidance to the industry, biopharmaceutical industry, it is clear that how important to HOS characterization in order to ensuring the biosimilarity in the biosimilars to the biologic or innovators. So now moving to the in this slide, Nandisha already told about the primary structural characterization, including study of intact subunit mass, peptide mapping, peptide sequencing, disulfide mapping released in and O glycans, native mass, charge heterosity, which we execute in our facility using high-resolution mass spectrometry and capillary electrophoresis. Considering HOS characterization, we perform analysis related to secondary structure, tertiary structure, protein conformers, protein stability, protein dynamics, and particle size determination with the help of CD, DSC, ACC, FFF MALS, FTIR spectroscopy, and CD spectroscopy. Not only this, in our facility, we offer studies on charge variant, native map, intact map characterization, peptide metabolites, and oligos characterization using HPLC techniques and chip technologies as well. So this is all from my side. Now I would like to call upon Bhavika for the next segment.
Okay. So moving on, we would be going ahead with our question and answer session. We have some great questions presented here by our audience. The first question is, what is the role of high resolution mass spectrometry in precise molecular characterization? Mr. Nandisha, can you please answer that question?
Yes, looks good these questions. So as I mentioned, like higher resolution mass spectrometry in precision molecular characterization. What is the importance? Like as we know, high resolution mass spectrometry plays a crucial role in the precise molecular characterization by enabling the identification, quantification, and structural analysis of the molecule with exceptional accuracy. So when we'll do all this, when we say high resolution mass spectrometry, accuracy is matters a lot, especially for the molecular weight. So that is the one reason. And HRMS provides highly detailed mass spectra that allow scientists to determine the exact mass of the molecule, which is essential for distinguish between the compound and similar structures or molecular weight. Means any glycoforms or any other charge variants, anything is available by achieving high resolution mass spectrometry resolution. So HRMS can also resolve the isotopic pattern and detect the susceptible differences in molecular composition and offer insight into the complex mixture. So this level of precision is especially important in fields such as drug development, proteomics, metabolomics, and environmental science. So where accurate molecular identification and structural elucidations are play a vital role for understanding the molecular function, interaction, and mechanisms. Moreover, HRMS is vital in detecting the unknown compounds and confirming the purity and authenticity of the synthesized molecule. From this, I hope this will be fine.
Thank you. Thank you. My second question is directed to Dr. Shoman Shaha. What are the key strategies for biosimilar development from an analytical characterization perspective?
Thanks, Bhavika, for the question. From an analytical characterization perspective, key strategies for biosimilar development focuses on ensuring that the biosimilar molecules closely resembles the reference biologic in terms of structure, function, and performance. These strategies involves a combination of techniques designed to assess the compare critical quality attributes for the biosimilar and the reference product. These key strategies are including comprehensive structural characterization, which includes amino acid sequence to confirm the sequence of biosimilar protein, peptide mapping to identify the post-translational modification like glycosylation, phosphorylation, oxidation, and mass spectrometry to accurately measure the molecular weight of the biosimilar and identify any differences in structure or modifications. Coming to the HOS part, technologies like circular dichroism, fluorescence spectroscopy, this study tells us about the secondary and tertiary structure of the biosimilar and how much accurate with the biologic molecule, and differential scanning, calorimetry, and dynamic light scattering may also use to access the thermal stability and particle size distribution. So this is how we'll strategize.
Thank you. My third question is what are the key challenges in the analytical characterization of complex biopharmaceutical? Mr. Nandisha, sir, can you please answer that?
Yes, thanks for the questions. The analytical characterization of complex biomolecules faces several challenges due to their structure complexity and variability. So these are from this now we can I can tell the few highlighted challenges. Well, this includes like structure complexity and heterogeneity. Means like if the as we know biopharmaceuticals such as proteins, monoclonal antibodies exhibit complex in structures, so and the variations, so that making accurate characterization difficulties. So this is like mainly the structure and complexity heterogeneity plays a main role challenges while analyzing that characterization of this biopharmaceuticals. And next one is like post translation modifications, whether any post translation modifications is occur during that uh, processing or any movement, all the such as like for example take as a glycosylation, which complicate their analysis requiring the advanced techniques. So like this is like another thing. And then size and conformation analysis. So when it is come to size and conformation analysis, whether that molecule is like a large size, large complex molecules requires a precise methods to access the structural changes and aggregation studies. Example, if you do HRMS, which will give that precise methods for access the structures, and if there is any aggregation studies are there that we can do with SEC things, which will give like what is the percentage of like monomer, dimer, tetramer, all these things. And then when we'll do this like complex biopharmaceutical characterizations, so the we need to do the batch to batch variability. Like, means like we need to compare the things, so that time the differences in manufacture conditions can lead the variability and making the consistent quality control in challengings. So and then one more thing is like most important, that is like detection of impurities and aggregates. So like what is the percentage level of like impurities it should be available to identification of the death, identification of the impurities, and then aggregates. So what is like aggregates are available, this is like aggregates are like technically demanding things, like how to check deal with the aggregates. That is the things after this five technical things. And last and most important thing is like regulatory compliance, whether meeting straightening the regulatory requirement for safety, efficacy, and consistency, add the complexity to that characterization process. So overall, advanced techniques and strategies are necessary to address these challenges and ensure the quality and safety of the biopharmaceuticals. I hope this will find the answers for the questions. If any still question is there, you guys can type it in QA session. Hello.
Thank you. My next question is, what are the emerging technologies transforming biosimilar development? Dr. Showman, can you please answer that question?
Oh, I'd like to say it's a nice question. As science is getting advanced, we are also adopting the new techniques which are helping us every day. So, emerging technologies which are enhancing biosimilar developments in several ways, and we are adopting that. So, so I'd say some of start with hydrogen deuterium exchange mass spectrometry. We call it HDX-MS, which is new technique that has emerged as a powerful tool for studying protein conformation and dynamics. This technique measures the exchange of hydrogen atoms with deuterium in proteins, providing information on folding stability and structural differences between the biosimilars and the reference biologics. Another technique is fluorescence detected circular dichroism, FDCD. This technique combines circular dichroism with fluorescence to provide insights to protein folding stability and tertiary structure. FDCD allows for more precise measurement of conformational change that might arise during biosimilar development or storage. Another emerging technique I would like to tell about 2D LC, like two-dimensional liquid chromatography. Recent advancement in the chromatography techniques, including 2D LC, allows for enhanced separation and detailed analysis of complex protein mixtures. These systems are crucial for detecting charge variants, glycosylation patterns, and other PTMs that are important for biosimilar characterization. These technologies improve the efficacy, quality, and stability of the biosimilar development. So these are few, so many techniques are there. I just told few of them. That's all.
Thank you for answering most of our audience's questions. But I would like to take a pause over here. This session has indeed kept us all engaged, but we also are having certain time constraint. But please be rest assured, we will be answering to all your questions. We'll be emailing Dr. Showman, Mr. Nandisha will be answering you the questions, and we will be emailing them to your emails. And also we will be sending a recording, this webinar's recording, to all our registered participants. Lastly, I would like to earnestly thank to all our participants, all our attendees. Thank you, Dr. Showman. Thank you, Mr. Nandisha, for being such wonderful presenters. Thank you to our audience for tuning into this webinar, taking out the valuable time. Till then, stay safe, stay healthy. We hope to get you again in our next webinar. Thank you.
Thank you. Thank you, everyone.
Thank you, Bhavika. Thank you, everyone.
Thank you.